Welcome to the fourth day, and hope you're all doing. From today, we are going to transition to a little bit more evolution, and actually, and tomorrow, actually, a bunch of research talks also from very diverse. So we have also, first we'll start with Jahnavi, but then we have Ashwin Seshasai, who works on evolution in microbes, molecular biology and evolution in microbes, and also Niaz Ahmed, who works also on specific pathogenic microbes, right? Who else today? Ansil? That's all today, right? Huh? Oh, Ranjit is also today, right? Today or tomorrow? Oh, okay, great. And Ranjit, who's here from Bangalore, also works on population genetics, and he's going to give a talk about his work on COVID, right? Yeah. And then tomorrow is also is going to be pathogen genomics, mostly through the day. So, and then we'll transition to virology and all that stuff. So this disease transition is happening slowly. So very happy to introduce Jahnavi Joshi. She's a faculty at CSIR CCMB. And she started her journey in research with an interest in centipedes and biogeography. She's done some really wonderful work, which Robin referred to in his talk yesterday. And today she's going to do more of a conceptual lecture about coevolution, because this is something she's been working on. Recently, she's been working on venoms in these centipedes. So she's been thinking a lot about coevolution of venoms with species. So Jahnavi.
Thank you, Uma. Thank you so much. I really wish I think more about coevolution and centipedes and venom evolution. But what today I thought I'll do is that we are a small group based out of CCMB in Hyderabad, and that's the lab. And as Uma said, last 3 days, I think you guys have thinking about ecology a lot, about host and pathogens from ecological perspective, and you're shifting to evolutionary. And that's precisely our area of interest. We ask questions in ecology and evolution. And that's why our lab we call evolutionary ecology, because we dabble in both. We either ask an ecological question or evolutionary question and use approaches from both fields. Predominantly, we work in these 3 themes. We try and understand the biodiversity around us using integrative approach, where we use morphology, molecules, the climate in which species live, get our species hypothesis in place, and then try and understand how they have diversified, speciated in the given landscape, and ultimately how this forms communities. And I think, and then ultimately how communities are formed. So this is the broad area of the research. Today, for next 1 hour or so, what we thought we'll do, and I really want you guys to contribute to this, is think about how do you think about host and a pathogen, because I think that's what many of you are interested in, and what the consequence, do they really co-evolve, and what are the consequences when there is a coevolution which happens, and how does one go about studying this? And this is a classic definition, and people have now gone beyond this, but this is one of the classic definitions which says there has to be a reciprocal selection, an evolutionary change in a species A when it dictates a change in species B, and species B also affects species A. This is where you call reciprocal selection, and this is a classic definition. People have now come up with more variation of this, and say some people have even argued that reciprocal selection need not be always the case for a coevolution to occur. But if you still look for textbook definition, how one can define coevolution, reciprocal selection is one of the key element in thinking about coevolution. What else? I think yesterday Ashwin referred to a hypothesis which is also very much in the field when we think about coevolution. Anybody? So same idea, same concept has been formalized as a hypothesis. All of you have heard it. Red Queen hypothesis. What does it say?
Someone?
So continuous interaction between the species, and which pressurize themselves among themselves and see that in the best way what is the best way out of, even after so much of friction, whatever the adaptive level which supports for its survival.
Okay. Any other thoughts? What Red Queen hypothesis says? Evolutionary arms race, okay. So evolution is occurring continuously, species evolve continuously, and their relative fitness, how they affect each other, their counterparts, who the species are interacting with, is the study of, like that would drive the coevolution, right? And somebody already said evolutionary arms race. So the species which interact can benefit each other or can have an antagonistic interaction. When there is a beneficial interaction, we call mutualism. If there is an antagonistic interaction, it could be parasitic interaction, right? Or host-pathogen interaction. There is an interaction. It can have a positive interactions or you can have a negative interaction. And host-pathogen interaction is one of the negative kind of interactions. And people have been looking at these interactions in a multiple frameworks. So for anything to happen, when we are saying these are evolutionary interactions, what are the time scales come to our mind? Where we are saying, okay, species evolve continuously, they affect each other's fitness or relative fitness. How do we study that?
So, sorry, you're saying these are all distinct.
Not distinct. There are different versions of the similar interactions, and outcomes, it could be either positive interaction or a negative interaction. It could be a single species, it could be a group of species which interact with each other, lead to a coevolutionary pattern. What are so you can think about mutualisms, which I think pollination, seed dispersal, these are all called mutualistic interactions. Both species or interacting partners are benefiting. Versus host pathogen may not, like for their survival, yes, important, but the way we see that interaction, we think it's a negative interaction. You can have a competition where species evolve traits to use any resource, be a food resource, mate resource, habitat resource, which is differing, which can also lead to a coevolution. And then you have a parasitism or a predation where one species benefits at the expense of other. And I think most of your host pathogen interaction will form in that category, correct? And in the literature or a textbook knowledge of our understanding of coevolution, it can be a species level, it can be a group level, or it can be at organelle level. And this is from a review where they have listed all examples of coevolutionary things. And these are very distinct groups of organisms which are evolving with each other. Would you agree that these are all examples of coevolution?
Coevolution.
Yeah, these are, and what are the implications? What are the consequences of those interactions? And those could be very, very different. Yeah, and where would then now host parasite fit in? This larger. So now are we thinking about any host and a parasite? It could be a mammalian host and arthropod parasite, it could be a nematode, it can be very distinct life cycles, very distinct life histories, and now we are thinking about how would they influence each other.
So I just want to ask or maybe understand and clarify is, so when you are saying the consequences of those interactions, like mitochondria with eukaryotes and bacteria, so isn't that kind of a selection and processes are happening because that's what is needed? I mean, there's a kind of a selection pressure going on too, and that's why those bacteria are digesting those kind of things, right?
Yes, yes. So selection is operating. And when we think of coevolution, we are thinking about selection, right? We are thinking about their relative fitness because how you interact with any other organism or a group of organism, you are influencing each other's relative fitness. So of course, there is definitely selection at play, and you have stable coevolution happened and it has retained. But do we agree that these are all examples of coevolution, and host-parasite form are part of this bigger umbrella of coevolutionary studies, and that's a negative interaction, and that can happen with very completely different kinds of organisms can interact with each other and influence each other's evolution. And one of the consequences for these interactions is for their reproduction. So when I have to think about if it's co-evolving or not, I need to think about what's the consequence of that interaction. I can then think about, okay, these are the consequence for their reproduction. So reproductive fitness could be something can be then measured, right? I need some consequence to that. I can measure those consequences.
In taxa 1, there is a new for mitochondria eukaryotes, and taxon 2, it is a bacteria. Does it bacteria, there is a theory, that endosymbiotic theory, where machloroplast in mitochondria, that previously it was an unicellular organism that were prokaryotes. Is that relating to that or what?
No, this is, what's the origin? Like you have now mitochondria coming as a whatever growth and now part of eukaryotes. Has it happened because of coevolution? Is only that.
Okay.
So what process which has led for a mitochondria to come into eukaryotes?
Eukaryotes. That's true.
And coevolution can be studied at two scales, right? We can think about a short term and a long term when we have to define now how we will study it. And I think, I don't know if you guys have already discussed short term coevolution, which will be at a population levels, right? You will think about reciprocal selection pressure resulting in more at a population level processes. And one of the example could be, I can think about how allele frequencies change in a population. If these two interacting organisms, taxa A and taxa B, interact with each other and they are affecting each other's fitness, I can think about changes in allele frequency as an example, but I can think about many other things. But those processes I will look at a small evolutionary time scales, at a population level. But when we think about long time evolutionary time scales, we can then think about coevolution leading to co-speciation, right? Because over millions of years, if two species or two groups of organisms are affecting each other's fitness, will lead to a distinct species. And does that happen? As a result, what we will see is a co-speciation. But is this agreeable? Do you have other thoughts on this? Yeah, so the rest of the talk, what I am going to focus on, not much on the short-term consequences of coevolutionary processes, but a long-term consequences of coevolutionary processes, and does that lead to a co-speciation? How frequent is it in the literature that we see co-speciation has occurred, and is it easy to study that? You can use different ways to study that, both short-term as well as long-term. Today we will focus on how to use phylogenetic thinking to study co-speciation. You can have field observation and experiments which show you that coevolution is happening. You can have a lab-based control experiments to show that coevolution is happening. You can have computational simulation models which show that there is a potential for coevolution or coevolution happens and then these are the outcomes for it. Or you can, to study, reconstruct the past and think, whatever we see today, is it a product of coevolution? And one such outcome is co-speciation. And most of the rest of the talk, what we will talk about is how do we use phylogenetic trees to think about coevolution. How many of you have heard or reconstructed or used trees in your work? How many of you are familiar with this term, molecular phylogeny? Can you tell what does it depict or what does it tell us? If I see a tree, what does it show or what does it? Distance between the species.
we're all scientists, right? We're all scientists. One of the important things in science is precise words, speaking very precisely, right? So just
So distance between two species, like relatedness between two species.
Okay. And the relationships. Okay.
what is the difference between distance between two species and the relationship? Two things which have come up. Anyone? Okay, origin.
Common ancestry. Okay.
The evolutionary distance between two species is how far they are having a common ancestors and how they have evolved. And the relation suggests how two species are related to each other in evolutionary timescale, what common ancestors do share.
Great. Yeah. So just the visual depiction. You have a common ancestral lineage which splits into two. This process is ongoing, right? Some lineages go extinct. What we see today as a diversity is this we call tips. The distances or how two species are, how far are they from their most common recent ancestor is depicted by these branches. We call them, these are nodes, these are tips, these are branch lengths, where the distance is, be it whatever measure you use to calculate that distance. And these are the nodes, which are ancestral nodes, right? And this is what we have today when we reconstruct tree, which tells us how these, whatever group of organism I'm working on, how the species are related to each other by shared evolutionary history. So common ancestry is the fundamental principle which we use here. Of course, what other information one can get when we are using molecular phylogenies is that time, divergence time. How long ago all these divergences have happened. So that's an additional layer of information which we can gather from these trees. Then you can use these trees to ask many ecological or evolutionary questions. We are interested in understanding how co-speciation has evolved. Once you have a tree, you can ask multiple questions. Today's focus will do co-speciation. And this is again a very classic textbook example. You have a rodent and its lice, which is a parasite on that. Now I want to understand if there are two species of rodents, do they have two species of lice, right? And is it because of the interaction of that parasite with respect to the host? You can have two species of gophers occupying two species of lice by randomly, or is it actually affecting each other? So whenever we are going to say co-speciation, we really want to assess, is it interacting species? Do they mirror each other, right? And how has this happened? And you can now think, is it species to species interaction, or is it at a lineage level where there are multiple species which interact with single parasite species? But the aim here is to think about, does this happen? How often does this happen? Do we have real examples where species to species there is a coevolution happening? And how do we study this? And I think Ashwin showed us yesterday quite a bit of trees where there is a host and there is a parasite, right? And if the interactions are so simple, you hardly get a real life scenario where you have one host and one parasite, right? It's going to be complicated, but what is the principle through which we can study this? Is it one-on-one interaction or one host, three parasite species, or vice versa?
So, Jahnavi, how can you say this is co-speciation? Maybe just by chance, there are 5 species of vectors and 5 species of hosts.
Precisely, I think what we will do in next 30 minutes or so, is it by chance, by random events, you have 5 species of this which are mapped onto this, or is it evolved as a response to your host, or host has evolved as a response to the parasitic interaction? But this is the concept, and whatever we are going to discuss next half an hour is going to be something to do with this. Is this, but you see there is a merit in doing this, because we have a host You have a parasite. I can have another parasite lineage here, which is not a lice, but a parasite which is on these lice. And it gets complicated if you keep thinking about who are the interacting partners. So we also have to figure out if this is the most important, significant interacting partner which is influencing the speciation. This is just a one way to think about it. So what do these co-phylogenetic methods allow you to do is, you have your host tree which is in the black lines here and a parasite which is red. You have it, you have to overlay and think, does it match with your host parasitic tree? If I do that, okay, I have a host A, host B, parasite B, and it matches completely with what I showed earlier here. 1 on 1, fine. Another information what phylogenies give us is time. Hosts have evolved 50 million years ago. Your lice have evolved 5 million years ago. Do I still see the similar pattern? So can I now use information of time? Does it happen at the same time when host is evolving? Or can you have infections which are much later, your parasites have come into the picture much later, then it's not necessarily a co-speciation. Right? This is not a response. What you have is a species which is your host, or they mirror the parasite. And parasites actually have evolved as a response to the host, but host hasn't evolved as a response to the parasites. Does this make sense? You can use the time information when these events have happened. And we'll come back to those events what we are referring to. But time as an axis is the information we can use. What we are trying to do here, now think about the events where it's not a one-to-one map. You have A, B, C here, A, B, C here, but the parasite tree is very different than your host tree. Then you have to think, where are these shifts from lineage 1 to lineage B or lineage C? And can I reconstruct these shifts? So red arrows are showing host shift. A, B, C, D, E, F, G, H. If J shifts to C as a host, what should C here? J and C, right? Does this happen? Do species shift hosts and then they evolve? Or they evolve in the same host? Sure. See, right now we don't have names here. We are just thinking these are the parasites. I give some names to these species. I have these 3 species A, B and C. However, these 3 species have come because there was a host shift from this slice to here. So these 2 will become more closely related as compared to right now what I have seen here. Because this lineage has shifted its host instead of the sister host and speciating here, it shifted and it came here. Yeah, and I think we have more examples for that. Is different. Yes, we'll come to that in a minute. Yes. What you can also see here right now, you may have a same set of species. Here your time tree also matched with the host tree. However, here parasites show much younger divergences, which is correct. You have parasite which has evolved. We'll come to that, how to use time in this. Right now we are just looking at topologies. Then we'll come to the time where you have a lineage which is evolving much at a shorter time with high rate of evolution as compared to your host. You can have complete discordance. It doesn't need to be because of coevolution. You have a host and a parasite. Yes, you have one host, one parasite lineage, but they don't have a mirrored evolutionary histories. Or you can have multiple parasite lineages which are also closely related, which are evolving in this host. And then you can again think about how has that happened. And this is, I think, which is what people find in the empirical systems, that it's not a single host lineage. You have multiple parasite lineages which are evolving with respect to your host, and you will have that evolution. So we'll come back to it, which are these conflicting nodes and why they are so, but you could have scenarios like this. So you can have a complete one-on-one matching, you can have host switch alone, you can have a divergence times which are different, you can have a complete discordance, but still you have a species to species interaction. But it's not a product of co-evolution. You have one host, one parasite, but these are very distinctly related parasites. They are not because of co-evolution. There's still species specific interactions, but they are not product of co-evolution. Or you have multiple closely related of parasite lineages.
You are saying that there is the coevolution between the parasite and the host is independent.
Yes, they're not happening it together. Like it's not because, so for example, if you have a lineage here, yes,
Yeah.
this is your host. You have now 3 species of parasites. Let's say these are unique, but they are not sister to each other. What you have so your A, B and C is not sister to each other like this.
Okay.
Yeah?
That means coevolution within that species is independent.
Yes.
Or
these
not
things have
between the host.
assembled in the space. Right now you have interacting species which are one-on-one. But their evolutionary history doesn't tell you that they are sister species. They are responding to similarly how hosts have.
Is there any relationship, the pressure, evolutionary pressure
Need
on
not be.
host
That you have
due
to
to
figure
because
out.
of
But this is
parasite?
not because of the co-evolution. It's not the parasite which is evolving. It's not a reciprocal selection which is leading to that.
Maybe I'm wrong, but if there is a lot of selection, so when you say pressure, how does that impact a phylogenetic tree? What we are measuring, right? Sorry, I might be wrong, but basically selection would result in shrinking or expanding of those branches. That is, very big differences or not much difference at all.
Yeah, or we can just think about in this way that what has happened, these lineages are not here to occupy these hosts because they are in some other place, geographical space, or they occupy different hosts. So there is not host specificity. You can have a host switch events, and which will come to that in a minute. These could be because of result of host switching events where there is still a weak coevolutionary signal. But now, for example, D, E, F, these are not in part of ABC, which is your host, but very, very distantly related host. So it's not a response. So one of the fundamental definitions will go back to it to say that it's a co-speciation. It has to have a reciprocal effect, which is not the case here. There could be other selection pressures which are operating, but it's not a reciprocal selection issue where host interacts with a single parasitic species and a parasitic species is responding to that selection pressure. Do you want to?
There are a couple of questions here, and then.
Yeah, I was thinking, what is the possibility of misinterpreting this if the hosts in that tree, if many lineages have gone extinct?
Yes.
Then
we'll come to that. We model extinctions
Okay,
there.
yeah, thanks.
There is a risk, and one has to model that extinction risk also.
So can I? So Jahnavi, say I go in the field, like Ashwin also showed lots of phylogeny, and then we are looking at the host and also the parasite or whatever we are looking at. So what I'm trying to say is that all phylogenies are very subjective, right? I mean, if you keep adding lineages, then only it gives you a proper picture of what's related to whom. So say if I have done only those ABC and ABC, I would say, wow, these species are actually kind of co-evolving, but that larger picture is hidden somewhere else, basically. So in order to tease that apart, it would be nice if you...
Yes. Yeah, and thanks, Farah. That's a very important point. Having a robust phylogeny and a complete taxon sampling is very, very important. We can't selectively, and she pointed out a precise problem here. If I don't have a parasite phylogeny, which is a complete phylogeny, I'll completely miss, and I'll have only these species where again I can reconstruct the tree like this. And I have missed out on all the other lineages, which are because I haven't sampled those lineages. So whenever we do this, we have to think about complete taxon sampling. Like from host perspective also, I can also have a host only which is A and C and B is missing, or more diversity of parasites. So the sampling has to be done adequately for both. What are the measures of this? Having a stable tree topology. So trees have two values you can think about if it's a stable relationship. Each node, we calculate something called support. How confident are we that this is the real scenario? And we call it hypothesis only, but you will have some support values to these nodes to say that, okay, this is the relationship with the confidence of this. So there are two ways. If I use a bootstrapping method in a likelihood or a parsimony framework, I get number 90%, 60%, 70%. If I use a Bayesian approach, I run MCMC chains and I get a posterior probability value. Is it 0.9? Is it 0.8? And then you can think about how stable is your tree topology. If your tree topology itself is not stable, you can't do any of this analysis, and that's the very fundamental to any of this. You have to have a complete taxon sampling. Having a stable tree topology is very important. Yeah.
Hello. So what if this in the case of obligate parasites? So they required a host for their survival. Without the host, they can't survive, right? So how can you give this phylogeny for that obligate parasites?
What do you mean?
Obligate parasites.
Yes.
So does they evolve coevolutionally?
So we'll test if it's obligate parasite and is that a relationship which has an impact on co-speciation or not. So you will design, you will look at how many hosts does it have, 5 species, 10 species, or whatever the host species are, and all the obligate parasites, how have they evolved?
Ma'am, just you are mentioning about likelihood, maximum likelihood approach and parsimony method. So how do you, know when to use a parsimony method and when to use this maximum likelihood method?
Depends, but I think understanding is parsimony has its own limitations. So use a likelihood or Bayesian, and we'll discuss parsimony method to reconstruct this tree, but it's still useful as an exploratory method to get to understand what is happening. The principle parsimony uses is evolutionary events. If you are building a tree, for example, how many nucleotides have been substituted? What's the minimum number or minimum steps I can say to get to that tree topology? So that's a different philosophy or a principle it uses, versus likelihood uses a different principle. So you can do both and assess the tree topology, but these are two different ways of building tree topologies. Parsimony has a limitation because you can't calculate divergence time. So that's why we have to use a likelihood approach. And likelihood or Bayesian are very similar in terms of the way we use data. So if you can have a parsimony tree as well as a likelihood tree, but there are limitations of a parsimony method where you can't do the divergence time dating, which you can do only with a likelihood tree.
Okay, thank you.
Is there a question? Can we move? So there are two ways we can see. We can just do a rather field. In this field, people have looked at just the tree topologies and looked at is it a coevolution, or some have actually tried to model these events or processes to see if is a product of coevolution. So the first set of methods which use tree topology, and I think Thanks to our earlier discussion, we can see what it essentially uses is the topology. Topology means relationships. What A, B, and C are related to each other and your parasite. So I have a topology of a host, I have a topology for parasite. Aim here is to see if they are congruent. Are they similar? Why will they be similar? If there is a reciprocal selection, our hypothesis is if there is a reciprocal selection, they are interacting with each other and exerting selection pressure in a way that they are responding to each other, and you will have a mirror phylogeny. So is there a congruence between these two tree topologies? So those are called topology based methods. There is a different, I think some 4 or 5 different kinds of methods looking at topology. So basically the idea here is that look at the probability of observing certain level of congruence with respect to your expected tree. Uma asked us, how do we know that these, first of all, if it's a product of coevolution is a secondary. If these two tree topology are congruent or not is the first step. And I think most topology based methods do that. So one of them is called congruence index, where your data set which is required to perform this test is a tree topology. Here, it's just the topology. These branch lengths are not incorporated, it's just the topology. So divergence time or a branch length rate of evolutions are not considered. Just that, does this ABC match with other ABC? Yeah. So branch length which tells us the rate of evolution or a divergence times are not included. You just use the relationships. Yeah. Again, in this method, there are no random trees generated to see if this is our observed and this is the random. Is this significantly different or not? It works only for one-on-one correspondence where you have host and parasite tips. But this is a useful exploratory analysis. Once you do this, you understand what is the structure of your data. It's not a confirmatory test, it's an exploratory analysis to figure out in your data set. You also have something called parafit, tree map, and those kind of methods where now you can incorporate branch lengths, you can have distance-based methods to do that, and either I can give a tree topology and use this branch length or convert this tree into a distance matrix. I can convert this distance matrix, and then what I have is 2 distance matrices, one for the host, one for the parasite, and then run a mantle test to see if they are congruent. Yeah, I can use a sequence alignment also if I don't have a tree topology, because I can get distances from sequence alignment also, right? So these are the methods which are largely used, either a sequence alignment or your tree topology, where you can incorporate distances. And it's now you can also not restricted to one-on-one. I can have a different topologies or different matrices. So if I have a conflicting tree topology which says I'm not so sure what are the host or parasite relationships. This is one tree topology. The other one is this. This relationship, this node is not supported. I have multiple tree topologies for a host or a parasite. Then I can use those also. I can also randomly generate for a given number of taxa how many tree topologies are possible. Then what you have seen is your host as well as your parasite tree, and see if it significantly matches.
If there are one too many connections between host and parasite, then how would you use the matrix method? What will be the indices in the matrix method?
So basically you constantly compare with one host.
Oh, okay. You take the
number
dominant.
Of pair of comparisons will reduce.
You take the strongest relationship and then you do
No,
it.
not strongest. Wherever is the possible. Like, for example, this is your parasite and a host. You all have B here. and only different species here.
Yeah.
All of these interact, like your one host has 3 parasite lineages. Is that what you're asking?
Yeah, yeah.
So now your
Or the
comparison
other way
be
also.
with all 3.
Or the other way also.
Yes, it could be other way, but
Yeah.
you are creating this pairwise distance matrix,
Yeah,
right?
and
So it
then-
doesn't really matter. It will have the same value for those many columns. Right? Your matrix is this. So A can interact with B and C and D similarly or differently. So you will have one distance matrix for your host
And
tree.
one you get for the other, then
you
you
will-
compare both matrices, right?
Yes, so you will have multiple such matrices, right? Which you will compare and then see where you see the same.
Okay, got it.
And I think if I understand you correctly, this is what the point is. You can actually think about how each lineage is contributing to that coevolution. If it's biased towards only one, like one host, 4 species, or is it one-on-one? So you can actually also think about which are the lineages which are incongruent and which are the lineage which are most congruent. You can identify those lineages and lineage level contribution to that whatever value you get. And again, this review, and Ansil, I can send it to you, all the literature, and you can circulate. So it has comparison of all the tests one can use based on these topology methods and what are its limitations. And again, people have started using Bayesian approach for this, but you still have to write to those authors to get the codes and methods. It's not available as a package yet. I tried looking for it, but I didn't see any. Sorry?
So this congruence index, are you comparing two matrices like what is their similarity measure or something?
So congruence index doesn't look at metrics. Congruence index is only t topologies.
Okay.
There is no index, there is no distance there. The other methods, parafit or tree map, actually uses distance matrix. And then you can use those distance matrices to look at either Pearson's correlation or mental test, whatever. These are your 2 distance matrices. Is that clear?
Jahnavi, I just want to maybe kind of clarify here right now. So when we are talking about distance here, this is basically genetic distance. You are looking at the substitution rate, basically how the ATCG or whatever is being considered. The other point I want to ask, or kind of would be nice to clarify, say, you mentioned the time thing, right? We do not have time information for lots of phylogenies, right? How do you go about that? Do you look for rate of substitution, like especially in the host parasite kind of evolution? How does that work then?
So we'll come to that when we think about event-based method, and parsimony is your best bet when you don't have a time information. But rate of evolution, we can again go back to sequence alignments and just look at what is the degree fold. And one of the examples which we will discuss has just looked at the substitution rates. What's the substitution rate in a host? What's the substitution rate in a parasite? By looking at your sequence alignments. Does that make sense? So time calibration is a different exercise. What essentially it's based on, no? So you have a tree topology like this. So I have a relationship which is based on the sequence alignment. I can use a model of sequence evolution, build this tree. I'm interested now, how many such substitutions per million year? How much time it would have taken for those differences to come about, right? So to do that, have you guys heard about molecular clock hypothesis?
Can you explain it once?
Yes. So see, you have a sequence alignment. Now I know that this base pair, it has changed. This is constant, this is constant, this is constant. Now by adding this sequence, there is also a base pair change here. I build a tree topology using this data. Now I know there are two mutations or two base pairs which have changed. Now I want to understand at what rate this change has happened. And the unit we use here is how many mutations or how many base pair changes have happened per million year. So how do I get this? The molecular hypothesis tells us that overall, this number of substitutions and time has a linear relationship. And we all understand, no, it's not a linear, there are deviations from this relationship. Can we model those deviations and use that to predict? If I have a fossil, for example, I say, I know this node, sorry, I'm just going to that. Okay, I say I have a fossil here. I know this could be 5 million years with plus or minus 2 million. And I have some distribution which I can assign to this. Can I predict now the other nodes based on number of changes which I see? That's the logic you use for divergence time calibration. Now I have a time axis here for all the nodes. So I extrapolate this information for those nodes. This is a very crude way of explaining divergence time estimation, but this is the logic for it. You can, if I know how a gene evolves, what's the rate which people have looked at across species, across taxa, we can use that substitution rate and say these many substitution per million year is the average rate. If I use that rate, what will be the time here? Yeah, I think Robin has extensively used molecular clock using SIDBI data, right? For birds where there are less fossils, you use substitution rates to calibrate time, but most accepted way or with your, if you want your credible intervals to be small, you can use both substitution rate and fossils and multiple fossils. So imagine this is your relationship. If I have multiple fossils to calibrate, the confidence will go up. And model all the deviations which are biological from this linear relationship. It could be generation time, substitution rates, anything. So model those.
Yeah, just want to add that some things don't fossilize well. So
Yes.
in which case, like maybe your parasites and things
Yes.
like that. So birds are like that. Some passerines, they have hollow bones. They don't fossilize too well. But some other birds do fossilize. So when possible, use fossil. It's like triangulating any kind of data, which comes out of a model. So, yeah.
So, one thing I think we should mention here, that molecular clock depends on absolute time scale, not the generation time. Because obviously parasite have a smaller lifespan and a human or any other host has a longer lifespan. So the amount of mutation accumulated over the lifespan or the generation time of human obviously will not the same for the parasite. So it has to be in a
Calibrated.
exactly, it has to be properly calibrated.
So all of these deviations, you use appropriate models for it to put it in your model for divergence time. And for the approach we use is a Bayesian approach. So you can model this. This is known information, right? Now we know generation time affects your substitution rate. For humans and parasites, it's going to be different. Can I model that? I know there are two fossils with this kind of uncertainty associated with. Can I model that? Can I use appropriate distribution for that and then model? Right? So all the deviations from this linear relationship can be modeled there. We know that extinctions also happen. So can I model those extinctions? So these are the parameters which will be in your model. So then you calculate divergence time. And some of these methods, why we started discussing this, can use dated time trees or can be used only the tree topologies, right, which don't have the time information. It's just the evolutionary relationships. But within that, you have two kinds of trees. One with a branch length or a parsimony method, which doesn't necessarily tell you branch length. You can use both. Some methods use that branch length information, some methods ignore. So now coming to the event-based methods, where we are actually interested now, is it a product of co-speciation or not? Right now we were looking at these two tree topologies and if they are congruent with each other or not, right? So this is again exploratory analysis one can call when you are just looking at congruence among tree topologies. You are not really saying this is the process which is leading to this congruence, but I'm interested in that. I want to understand, is it a simple congruence between tree topologies? This is the value, right? But to say that this is the process which is leading to this congruence, these are called event-based methods or co-phylogenetic methods, but which use some kind of a process you can invoke that process and then see if it fits your data. What are those? And I'm just giving you a example or a parallel here. You have a host phylogeny, you have a parasite phylogeny. How many of you have heard about biogeography analysis or done any biogeography analysis? Otherwise, it's not a great analogy to use here at all. But it's a very, very similar analysis in principle or a trait evolution model. I want to, somebody ask, how can I see this trait is evolved in this particular thing, right? So these are very similar in principle. All these models come under the same category. Host parasite, area, cladogram, trait evolution, they are in the same model categories. What you are essentially modeling here is a dispersal and vicariance. Here you are thinking about host switch and a co-speciation. Right, so these are the parallels, but if you haven't really done biogeography, let's just focus on this. So you have a host phylogenetic tree, you have a parasitic phylogenetic tree. My interest here is to think about if parasites have switched host on a tree and led to a co-speciation, or is it only co-speciation which is happening, which is leading to the tree topologies what we see today. Can I now model host switch, co-speciation, parasite speciation on host and parasite extinction or a host extinction using these tree topologies? No, so this is the analogy, but forget about it. If you haven't done the biogeography analysis right now, let's not go there. If any one of you had done that, this is a very similar analogy, that co-speciation is equivalent to vicariance, host switch is equivalent to dispersal, sympatric extinction, but forget about that part if you haven't done this. Let's just focus. So I'm interested in modeling these four processes on these two tree topologies. So that's the aim here. At this stage, geography is not in the place. Yeah, that is not something we are going to discuss because geography does play an important role, and in what sort of geographic settings and a mosaic these interactions are happening has a role, but let's not get to that right away. We'll just think you have a host, you have a parasite. How do I now invoke a process of host switching, co-speciation or extinction to understand how these three topologies have come about? So you have a host species, you have a parasite species. Imagine there is a congruence. My interest is how and when these host switch events have happened which have led to this, or co-speciation which has happened which has led to this. So I'm interested in identifying these processes on these tree topologies. One of the method which does this is tree fitter, and it uses something called Brooks parsimony approach, and it's a very, very simple and a straightforward approach to do that. All we are going to do is we are going to assign some cost to each of these events, host switching, extinction, or co-speciation, and then number of events, whichever number of steps are minimum, that model gets picked up. That's the principle of parsimony. And very, very simple approach is the Fitch-Wagner approach, and I'll try and see what do we mean by these simplest evolutionary steps or transitions, and how do we go about it. So imagine this is your tree. A, B, C, D, E are your host species, and these circles are your parasites. Right? And I'm interested what happens on their ancestral nodes, because that's where I'm invoking a process. Is it because of the host switch? Is it because of the co-speciation? Co-speciation means we had a common ancestor split into two. Same thing happened in the parasite tree. right? So what is happening on these nodes, or how do I think about these nodes using parsimony as a principle? And this is the, I think, most crude or simplest way to think about it. Imagine you have some states here. So I'm going to overlay these two tree topologies. You have A, B, C, D, E is your host and circles are your parasite. What will be here if I have a black circle, which is a species? Just also remember, I should have given some numbers. These are 5 different species. Imagine 4 and 5. What is the possibility of F having 4 species or a 5 species? What is the probability here for a G node of species 3 and species 4 and 5? So I'm going to reconstruct these states for all of these nodes. How do I do that? Extremely simplest way is I go from a tip to the node and then from node to the tip. It's called up pass and a down pass optimization. If I do that, I can go now from here. This open circle, H could be open circle. Again, this and this open circle could be open circle. Open circle and a black circle is equivocal or equal. We don't know what has happened. If I then have a final reconstruction which says open circle, open and black, then now I can go back of these two tree topologies and see, is it because of host switching? How has the black circle suddenly come? Or how is it retained open circle throughout the tree, which is a co-speciation event, right? In parsimony method, co-speciation doesn't get any cost. However, host switching is one as a cost, because you create a transition matrix for all of these nodes and then optimize it. This is a visual representation how optimization will be done, but what you are essentially doing is these tree topologies, you have some states, now you are reconstructing their ancestral nodes and saying, to get to that tip, what changes have happened at an ancestral node? Is it host switching, where black becomes open circle? Or is it just the open circle now into 2, which is a co-speciation, which has no cost associated with it? And this is a parsimony method of doing it. And I have one more visual for this. I hope there are different methods you can do, and TreeFit. We'll come to this in a minute. I think I have one more visualization for parsimony. We'll come back to this. Imagine this is your host and you have one parasite, and I have topologies. Now I want to see how parasites have evolved as a response to this particular host. So you have a...
What are the I, H, G and F? Are they the host as well or the parasites?
They are host only, but these are ancestral nodes. They are hypothetical ancestors which are leading to your A, B, C, D, E as today's host species.
And one more thing, in the downs, in the next slide, you mentioned it down, but on the G ancestral host, there is just a black circle, but when it goes to the up one, it becomes white.
Final.
Why is it so?
Because now you have 2 black, 1 open.
No, I just couldn't understand. You said that it is an up and down process from where
so
we
right
extract.
now if I go from bottom up, down to up, I'll do this, I and A, B and H, G and C, F and D. I do up pass, where I now come from here to down, and then a summary of this or whatever, a common between these is your final optimization. Just the same process, let's see, we'll come back to these things. Imagine you have one parasite, one host. Some barrier has come, because see, co-speciation can happen because there are barriers, right? Reproductive barriers to that. Host is also split, parasite, because host has split because of some barrier, parasite should eventually split and you have 2, which will reflect a topology like this. And this is called co-speciation. Is Is everyone with me? So these two depict two parasites. You started with one parasite, host 1. Host has shifted or whatever. There is a barrier now. There are parasite. Two parasite species now because there are two hosts there. S is a parasite, A is a host, A, B, whatever, those are coming. Again now there is a host gets split into two. Your parasite can eventually split into two. You have 3 parasites, 3 hosts. Is okay? Is because of co-speciation. However, we can now incorporate time into all of this, and we'll come to the time part of it in a minute. Another scenario, you have one host, one parasite. No parasites here. Host speciation has already occurred. Now these parasites are moving. This is called host switching. This is not happening along with the host phylogeny evolution. Are you understanding? And ABC can be related to each other or not related, but ABC are already existing. It's not happening together. Yeah? Then you have it this, and then it disperses or host switches into area C. Yes. Yeah. This is what? You can think about. This is a dispersal vicariance scenario. This is how it operates. Yeah, so it can keep on going. So now our aim is to figure out which of these processes which have led to this pattern, and we are seeing these true topologies. So what we are trying to figure out, which of these processes which have given to the tree topologies which I see today of host as well as a parasite. I can now incorporate information of time into this whole matrix. Yeah, I can now see, okay, this is how host phylogeny looks like. This is how the parasite phylogeny looks like. Now my aim is how S1 got to S2 and S3. Is it because of the host switching, or is it because of just the host also simultaneously was splitting, and it was because of co-speciation? But is everyone understood there are these two processes which we are thinking about? Host split associated with parasite split, or host switch. Host already exists, parasite has shifted to another host, colonized a new host. So we are now trying to model these two processes on these tree topologies. And that's precisely a job of these event-based methods, to see how many times co-speciation has happened, how many times host switching happened, and to make life complicated, we also have extinctions on both host as well as parasite. So how do we model those extinctions? Is this clear? But a simple principle here is what we are trying to model is how has that happened? Is it because of host switch, or is it because of co-speciation? Is everyone with me? So we were discussing these event-based methods, and parsimony is the simplest way to think about it, but we have already discussed what are the limitations. So co-speciation is the most parsimonious approach because it gives a zero cost. And we know that may not be the always case. In this framework, co-speciation cannot be costly at any point of time in this model. It's always zero, and host switching is a one cost. And doesn't incorporate divergence time because your tree topology just has branch topology relationships, but there is no information on branch lengths or a time which can be incorporated. And that's where the likelihood approach is useful, because now I can, when I'm reconstructing these ancestral traits here, I can also use the time taken to get to those 2 points. Of a host switch or a co-speciation, I can incorporate the information of time taken by both host and a parasite to evolve the way they have evolved. So branch length information, time information is used in a likelihood approach. And also likelihood doesn't necessarily say co-speciation is 0 and host switching is 1. So we can get rid of that cost matrix here completely. Here I can think about these as 2 different states, and create my transition probability matrix. So parsimony allows co-speciation to be always zero, saying that's the most common process which will lead to the tree topologies which we are seeing. Here we are not assuming that. We will create a transition probability matrix for both co-speciation and host switch, and where I can have an equal weight for both of these processes. And I can now think about extinction as a third process there. Because some of you have already asked, can we not have extinction events also? So we can also have now extinction as a process, and that framework allows me another process also to model. Parsimony doesn't allow you to do that. So that's why people have moved to likelihood approach. But parsimony is still useful to understand what's happening with your tree topologies. So to do that as an exercise, I would suggest that start with parsimony and then move to the likelihood, just to understand what is happening, what's the analysis you are doing, and then create your transition probability matrix for likelihood approach.
So my question was in regards to host switching again. So for example, if a parasite switches a host and its morphology, physiology is suitable for the new host, okay, and it has not evolved. It is exactly the same as it was in the first host. And might we think that this switch is fairly new because it has never evolved, because it was suitable for the new host as well?
So that's what the co-speciation would be. I agree with you. I hear you that it's the similar morphology, similar physiology as compared to where it's evolved from. So most common reset ancestor also has the same set of properties. However, now it's switched the host and it's in another host and it's speciated. What has it happened is created a reproductive barrier, right? Right now, this parasite is evolving in this particular host. Does it have a chance now to mate with the another host and that parasite? No. So the barrier has been created.
So the barrier would create irrespective of the primary pathogen or the parasite not evolving much in the newer host.
Then it will not establish itself, right? But if it's established with a similar properties as its ancestor or a sister species, what is maintaining that isolation is a reproductive barrier, because you are in a different host.
Okay, okay, because it has, okay.
Does that make sense?
Yeah.
Did everyone understood her question? That you have so she's asking a question where you are two species of parasites are in two different hosts. Let's use capital here. She's saying they look similar. Their physiology is similar. Maybe the mechanisms through which they infect is also similar. But they are in two different hosts now. Most likely all these characters which these two parasites have, also their common ancestor had. Right? But right now they are evolving in these two distinct lineages which don't mate and reproduce. So there is a reproductive barrier here. And what happens if I put A into B and B into A? And people have done that experiments. What happens? Do they establish? Do they infect? And what is the reproductive statistic? Hopefully one example has done that. So basically when we think about event-based methods, we are trying to understand which of these processes which have led to these tree topologies of host and a parasite. Is it because of host switching or co-speciation? And if I use a parsimony framework, I can think of these two processes. If I use likelihood, I can also model extinctions. I'm not going to discuss the likelihood approach here, but if any one of you are interested, I'll pass on the literature. So basically, you have your host, and my interest is here. On these nodes, which of these processes which have led to host switching, co-speciation, or extinction. And extinction can happen at both parasite as well as host. Likelihood approach allows, as we discussed, the time part of it, and also model extinctions, which parsimony doesn't allow you to do that. And this is something called instantaneous rate matrix, which can be created so that each of the process can have an equal value, and these weights are in our hands. We can decide the directionality, where parsimony you have to give co-speciation as 0 and any other process as 1. So in next 10-15 minutes, I'll just show you 2 examples in which these methods have been used, and these are examples of, I think, quite well studied birds. First one is a rodent and a lice, and the second one is a bird example. So these authors were interested in understanding, is it the lice and a gophers, the first co-speciation tree as a cartoon tree what we see, is it because of co-speciation? Are they really responding? So they sampled one of the rodent and its lice species in the US, and this goes back to the point which Farah asked us earlier. If you don't have a complete taxon sampling of a rodent as well as a lice, what happens there? So they went ahead, they looked at gophers. It's similar, very similar to what Ashwin told us yesterday. You go out, you collect host, species host DNA as well as parasite DNA. So you collect lice and you collect gophers. This is the likelihood tree of your parasite. And they have named these parasite species. Don't worry about the names, but you can see a same name with, so in a linear system, what you have a genus, species and a subspecies. So some of them have a very different subspecies. So this is called a species complex of your parasites. So they thought it's a one single species of a parasite, but they figured out it's not. There are multiple lineages of those parasites and they look like they are a new species. They even have to describe those parasite species in this case. So you can see there are some names which are similar, some names which are different. And all of these lineages then, this refers to the area in which they have sampled. So A1, 2, 3, 4, 5, 6, 7, 8. Each area, each population has a unique lineage of parasite. So they went ahead and looked at how many species of these parasites are there and then mapped. So, and then they do something which we discussed, the reconciliation analysis or congruence index or topology congruence part of it. They looked at a parasite and a host tree to see if they were congruent with each other. Yeah, so they have used mitochondrial and nuclear DNA to build a tree topology of your host. You have built a tree topology based on your lice. Looked at if they are congruent with each other. Yes, they are congruent. Then, which are these nodes which are not congruent? So what's happening there? Then they performed the event-based analysis to figure out how many nodes here showed us co-speciation event, how many nodes showed host switching events here, and what's the cost of that. And when they did that, they found there are 2 host switches, one loss, which is one extinction. So where are these? Now if I go and do the same exercise, I'll leave this tree with you. Remember we had a host and a parasite as a tip here. So you want water? You have parasites here. Think about all of these nodes now I have to reconstruct. What would be the ancestral node here? I know where there is a congruence between my parasite and a host, where these nodes show me that there is a host switch. These nodes are reconstructed on both of these topologies. Can you see where are the host switches which have happened and which nodes has that happened? Right now, they haven't used the dated trees. So it's a parsimony framework only, what has been used. You can tell me which ones and I'll point it out. 1, 3, 4. Yeah, see here, there is one. Where is the other one? You can tell me the number. Yes, 9 here. There is a host switch. Where else is the host switch? Yeah. Yeah, so you can actually, when you have these 2 tree topologies, exercise becomes easier to invoke a process now. Which is this process? How many co-speciation events have occurred there? How many host switch events have occurred to get to these two congruent tree topologies? And because if I have done the congruence test also using a method which also randomizes, so I have some kind of a statistical support for that congruence and identified which are these lineages which are incongruent and what is the process which is driving that incongruence. Yeah? So then I can see, is it a real example of co-speciation or not? Is everyone okay with this? Another thing we discussed, which was, do hosts and parasites evolve at the same rate?
So in the previous slide, you said there was a loss. So can you just show where it is?
Where the loss is? Which one you don't have? Which of these parasite species have not been able to colonize or remain the same species, didn't change into anything else? Can you see here? Yeah. Where there is no loss means also parasite has not infected that particular rodent species. So if I have a co-speciation happening, you have one host splits into two host species. Similarly, I should expect a co-speciation to occur in the lice, which hasn't happened. It's remained the same species and not two species of lice.
So the host should split, but the lice also.
Lice wouldn't. You have a same parasite.
Okay.
You can also have, somebody asked, can we look, I think Farah mentioned, can I just look at, because most parasite trees, you may not have time divergence data. Can I just look at substitution rates and think about at rate which host is evolving and a rate at which parasites are evolving? So these guys did that. They looked at, can we just look at how faster is host diverging and how fast or similar is parasite diverging? And when they looked at that, they found the parasite is evolving much higher rate as compared to host. And this was, I think, in 2007 or 8 study, so where they had access to the SIDB, mitochondrial data. Now you can have access to genomic data. You can actually calculate much more robust rate of evolution, substitution rates. Oh, no, these are just the hypothetical examples. We are just showing you what are the multiple scenarios you can have. When parasite evolves faster, what will happen? What equal rates both host and parasite have? What happens?
Diagram should be reversed maybe because the distance should be less.
Parasite is evolving faster, so you'll have a larger distances. Like evolution, what happens when you have more substitutions?
Okay.
Yeah, so if you have a less substitution. And host evolving faster, or you can have both. You can have synchronous, delayed. How does that look in a different framework? Yeah, so here they have used sequence alignments to look at what is the rate of evolution. And they show that lice had much more higher rate of accumulation as compared to the host, which was a rodent. So just to summarize, how does one even go about, if I want to set up a study to look at if it's a co-speciation or not, what are the important steps? And this is again from a review and which is in the reading material. You have to have host and a parasite trees, and all of these points one need to really think. What is the geographic distribution of a host? Have I sampled enough? Do I understand host pathogen specificity? And this is a background knowledge we have to have access to. Is it really a one parasite, one host? What's the host specificity? What is the ecological specificity? What are the feeding habits and environmental type? What are the life history traits of host as well as parasite? And we know that we really need to understand the species, because we can be discovering newer species there, and there could be species complexes. So you really need to think about if there are cryptic species, how do we understand taxonomic sampling, which is done completely or not? Is it a robust tree topology or not? Once you have this, both host and a parasite, then you can think about using any of these co-phylogenetic methods to assess both congruence as well as processes which are leading to these tree topologies. Again, in which you can use exploratory analysis, which is topology based largely, and then you can use event or a process based thinking if this is actually because of product of co-evolutionary events or not. And then you can then think about are these independent histories, right? Host and parasites are evolving independently, there is no interaction, there is no co-evolution per se. If it is shared histories, then now we can use both parametric models and divergence time of congruence and then test which are these co-evolutionary events which are leading to this co-speciation.
Can we only do this for co-evolution? This is slightly out of the topic, but can we, rather than looking at genetic evolution of one host, can we look at the subpopulations where the behavior is evolving, or some ecological or social characteristic is evolving and sort of overlay like this?
Yes, very well. People do it for biogeography, people do it for multiple traits. This is the similar framework one can apply to it. How is it really a response to one another? So A to B as well as B on A. So if there is a reciprocal selection, you can think about host parasite is one interaction, but you can think about mutualistic interaction, you can think about any behaviors which are, as well as here the unit is species, because we are interested in co-speciation. But you are asking, can I do it at a population level? Then my unit has to change. Then I can't do, maybe tree topologies is not the best way to think about it, but networks would be the better way to think about it. Networks can be the better way to think about it. But then the question, the unit has changed already, because right now I was thinking about more from a species perspective, that one species has a one host and I'm doing it. Now I'll have, if I'm studying a phenomena which is at a population level, multiple populations need to be looked at. But yes, similar framework can be used. I think we can stop here. I'll give these slides, but there's a very nice review done in 2013 to see how common is this. Do we see this co-evolution promote only the specialization, or it actually leads to a speciation? Because we can have a process where you have a divergence, but that doesn't necessarily lead to a co-speciation. So these authors actually looked at, if I'm not wrong, 60 studies or more than that, and they found the host shifts are common. However, many cases it's unclear, does it, so it's a large proportion of studies which support symbiotic divergence. So there is a divergence, but a small proportion which is actually co-speciation. And it could be a problem of data. We don't have the information of host as well as parasites. You don't have a complete taxon sampling. You don't have a complete phylogenetic trees. We don't have the other associated information. How is this interaction? Is it, what sort of specificity are we really talking about? How does parasite, what are the traits of a parasites which are needed for a host to respond, and what are the traits of host which influence parasite? So it could be still a lack of that kind of a data, or is it really a picture where the co-speciation events are rarer than people have assumed to be? But this was also a decade ago review. I couldn't look at one of the more recent reviews if there are, but this was a decade ago picture that co-speciation is still not very common, but co-divergences are much more common in the literature.
Rather than
co-evolution, like co-evolution leading to co-speciation.
So before that you told that we are adding costing of that.
Yes, so it will be co-speciation only, but you don't see the speciation happening. There is a divergence. We don't know if the result of that will be co-speciation or not.
Add cost for the more cost for the host shift, then our results will be the reverse maybe.
Host shift means?
You told the host shift we are adding costing more. So
Yes.
when
So
we'll
host
select
which
the,
has a cost 1 and a co-speciation 0. But if I use a likelihood framework where I
Okay.
don't assign the cost and both events are equally likely to occur, that itself will take care of that problem. Very quickly, another example where they showed, yes, there is this is between the pigeons or doves and their lice as a parasites. Again, you have a dove phylogeny and a host parasite phylogeny. There is a congruence. Most lineages have unique interacting parasite or host specificity, but here they went step ahead to think what are the traits which are driving this? What is this reinforcement which maintains this co-speciation? And they showed that the body size of a host and a parasite was correlated, and this could be one of the adaptive constraints. So they performed these experiments of shifting lice from a one host to another and see if they establish there. And they found the lice body size matters as well as the host body size matters. So that's where the body size and the mechanisms of feeding of lice, all those characteristics of parasites matter or traits of the parasites matter, and that's what is maintaining the host specificity. So you can combine the approaches of phylogenetic methods where you can show the congruence, you identify co-speciation events, you say this is what has happened, but now what are the processes which are driving that? So is it the parasite traits or a host trait? And in this case, it was both. The specific traits they looked at was the attachment. These lice, they have to attach to the feathers and then feed. Those traits, escape from the host defense and feeding were important, which were driving this host specificity. And that's why you see such a strong host specificity and a strong coevolution. But that's it. I'll stop here and I'll pass on these slides. And this is again a little old book, but which covers most of the phylogeny and co-speciation angle. It's called Tangled Trees by Roderick Page. So we have
China, suppose I'm just giving one example. China, one company was manufacturing SA1442 live attenuated vaccine against Japanese encephalitis in cells, baby hamster kidney cell line. And that vaccine was used in China. And vaccine recipients, because they behave differently. Like this live attenuated vaccine will behave differently based on their host, like where it is used, and some of the recipients, that virus was reverted back to wild type, and again it went to vector, and then in vector that virus was evolving. How I can prove that it is from the vaccine, like using sequence data?
So you will have a lineage which is from your vaccine, right? And this is the evolved lineage. If you build a tree topology, it should show you that it's, but you will need much more sequences to do that, to identify, is it a derived, whatever, the lineage which has evolved, what is the ancestral sequence from which it has now evolved into a whatever lineage. So what is the ancestral sequence of, I'm guessing, vaccine? And that may have come from some other virus. Origin, that virus, your ancestral, and now this. If you do that and use either a network method or a tree topology based methods, you can show what is the ancestral lineage, ancestral sequence, and this is a sequence which is evolved from this ancestor.
But how I can differentiate between wild type, because it was derived.
So you will have that sequence also, right? How the mutations have accumulated. What's the path through which the mutations have accumulated? So there is a suite of character evolution methods. Here your characters are your substitutions. How A has changed to T has changed to C, and when has that shift happened over a tree topology.
Okay.
Does that make sense? But you will need enough sequences of your wild type vaccine, as well as now it has evolved in a host again. if I understand that correctly.
Yes. Vaccine sequences from the host, like where, or vaccine
whatever is
manufactured or vaccine virus from the recipients.
If you can figure out both, I think both will be ideal. But if you have only one, is that one. Any other?
Yeah, if we try to figure out host and pathogen coevolution, we are taking a genomic sample and treating it to phylogenetic analysis. So the coevolutionary studies gives us if it is co-evolving. We are taking some available genetic sequence. Would it be better if we concentrate on a gene which causes virulence or compatibility and a congruent gene which causes?
Great question, and thanks for that. I should have mentioned this in the beginning. Your tree topologies are based on neutrally evolving markers. They are not the markers which are under selection. So it's not, if what you are asking is, if I look at a part of the genome which is under selection and it's part of a response to the immune response or anything else, those can then be studied using these tree topologies. However, phylogenetic relationships or evolutionary relationships of organisms, we use only neutrally evolving markers to base, because you are interested in how those organisms have evolved. And then you can map, overlay the immune genes or immune response on that.
So those responses may not be showing such similar.
Yes, of course. And that could be a question then. How have immune response evolved? But evolutionary trees, phylogenetic trees which depict evolutionary relationship between organisms and how have they evolved is always based on the neutral markers, neutrally evolving markers. Yeah?
How do we know that if a marker is like neutrally evolving or like is it under selection?
We discussed one hypothesis before. How do I test if the marker which I am using to reconstruct trees is neutrally evolving or is it under selection? Housekeeping genes, because they are under positive selection. Okay. What else? How do I test it? I have a data now. For example, I have looked at gene X in nuclear genome. I have sequence alignment of 1000 base pairs for 50 species. I want to understand if it's a neutrally evolving marker. Yeah, but what would be my expectation? If it's a neutrally evolving, what it should show. If it's not neutrally evolving, how should be the substitution rates?
Rate will be low.
Can I look at the ratios? Anyone else?
Can I answer?
Yeah, please.
Yeah, you can check the kind of substitution, synonymous and non-synonymous substitution, or if they are equal, you can assume that the gene is evolving in a neutrally.
Yeah, so we discussed one of the relationship. What was that relationship? If you are evolving, so when, yeah, I think you must have thought about null models or neutral models before. Yes, we also discussed linear relationship. Does it differ from that? What are the synonymous and non-synonymous? What are synonymous mutations and what are non-synonymous mutations? So do you know this? There are two kinds of changes, synonymous and non-synonymous. And why do we care if you have only non-synonymous or synonymous, and what information is that going to give us?
Synonymous mutation is there, then amino acid might be same, but nucleotide is changing.
Okay.
So that main function of the protein will be same, that will be not changing. But if nucleotide change and amino acid also change, then function will be changed. That's why we are more focusing on.
Yeah, and if we can then divide the number of substitutions we have in our alignment into synonymous and non-synonymous and look at their rates, right? Does that make sense to you? We segregate the mutations of synonymous and non-synonymous and look at their ratios. And can I test the neutrality of gene? That if this gene is evolving under neutral rate or not by using these data sets.
Magnum Kreitman test can also be useful here because you are looking at the divergence versus, so in case of synonymous substitution, we expect it is more happening in the population level. And in case of two species, we expect more divergence. So sometimes it's useful to do MK test, Magnum Kreitman test, instead of KAKS. KAKS is a broader version of this MK test, but it will give more specific answer.
Yeah, but if at all I don't use any statistical test, I just want to see how are the mutations spread across this and look at synonymous and non-synonymous mutations and then use any of the statistical tests.
If it is neutrally evolving, there is a chance of equal chance of synonymous and non-synonymous accumulations. And if it is selective
But you
pressure,
have more synonymous than non-synonymous mutations.
selective only synonymous.
And I can simply plot. I can plot number of mutations. When do they saturate? And there are different reasons for this saturation also. Let's not get into that. But I can just plot and see if they are still accumulating linearly or not, or they have saturated, right? Close, yes. Yes, I'm around whole day today. Yeah, thank you. Thank you, everyone.